Okay, let's unpack this. Today, we are diving into two of the most critical and, I think, really interconnected frontiers in modern longevity research. First, how we measure aging, and second, how we can maybe stop the engine of age-related inflammation, right at the cellular level. This isn't just about adding years anymore. It's about dramatically increasing health span.
It's a spectacular set of sources we have. They give us this holistic picture, moving from the macro, the big picture, all the way down to the micro.
Right.
For decades, we've struggled with this concept that aging is incredibly heterogeneous. I mean, your chronological age, how many birthdays you've had, is often completely irrelevant to the actual biological health of your individual organs.
Yeah, you could have the heart of a marathon runner, but the liver of, well, someone significantly older.
Exactly. And until recently, measuring that variability was difficult, invasive, or just way too expensive.
And that's why our mission today is so compelling. First, we are going to look at a pretty revolutionary system that uses standard blood tests, plasma proteomics, to track the specific biological age of 10 different major organs.
Then we drop down, way down,
Yeah.
into the nucleus. We'll be investigating the complex cellular survival circuit involving the famous p53 protein.
The guardian of the genome.
That's the one. And how does protein dictates whether a damaged cell initiates repair or flips the switch to become a toxic driver of systemic chronic inflammation.
So it's the story of tracking the decline and then finding the molecular switch to stop it.
Precisely.
Let's start with that big data approach then. The organ aging clocks. These are officially called proteomic aging clocks, or PACs. Walk us through how they work.
So it's machine learning applied to biology at scale. Researchers measured thousands of circulating proteins found in plasma from simple blood draws, and they taught an algorithm to predict age based on those protein signatures. But they didn't just stop at predicting your overall age. They trained it to calculate the biological age for 10 specific organs.
10. So which ones are we talking about?
The brain, heart, lung, your immune system, arteries, intestine, liver, kidney, muscle, and the pancreas. The big ones.
The validation for this must have been massive, right? You can't just throw out numbers like that based on a small study.
Absolutely. The rigor here is crucial. These clocks were developed in the massive UK Biobank. We're talking over 43,000 participants.
Wow.
And then critically, they were validated across diverse independent populations. In China and the US, this ensures the protein signatures aren't just, artifacts of one specific group of people. They reflect universal aging dynamics.
So the key metric they use is this age gap, which is the proteomic age, the one from the proteins minus your actual chronological age.
Exactly.
So if the gap is positive, your organ is aging faster than your peers. If it's negative, you've sort of hit the longevity lottery for that specific organ.
That's a perfect way to put it.
So what did the data reveal about the relationships between these different organ ages? Were they all in sync?
No, and that's the key finding. The data scientifically confirms what we've called the mosaic aging hypothesis for years. They found the age gaps for the different organs showed only weak to moderate correlations with each other.
How weak are we talking?
We're talking mean correlation coefficients between 0.10 and 0.19. So put simply, if your liver is 5 years older than your chronological age, that gives you very little predictive power about the state of your heart or your kidneys. Aging is truly organ-specific.
That distinction is huge. It means future anti-aging strategies can't be one size fits all. So if we can measure this specific organ decline, what's the predictive power for actual disease?
This is why the clocks matter far beyond just, curiosity. These pangers predicted future disease onset, progression, and even mortality above and beyond established clinical risk factors.
Even genetic risks.
Even genetic risk factors. They're like a real-time snapshot of your system's health. For instance, an accelerated kidney age gap was strongly associated with incident chronic kidney disease, CKD, and also the future development of type 2 diabetes.
So it's a targeted early warning system. Can you give us an idea of just how targeted it is?
Highly targeted. In 65% of the significant tests they ran, that's 121 out of 187 associations, accelerated aging in a specific organ predicted diseases affecting that exact system.
So heart aging predicted heart disease, liver aging predicted liver disease.
You got it. It's a clear, quantifiable link between biological aging and functional decline.
Okay, here's where it gets really interesting, though. When you look at all 10 of those clocks, one organ consistently stood out as the king of mortality prediction, and that was the brain.
Absolutely. The brain is the canary in the coal mine for systemic decline. The brain aging clock consistently showed the strongest overall associations with poor outcomes.
How strong are we talking?
We're talking profound. Brain aging was the single strongest predictor of all-cause mortality. It showed a 44% higher risk of death for every 1 standard deviation increase in its accelerated age.
44%.
And not just that. When they ran the statistics, the brain clock was independently associated with 14 of the 17 measured health outcomes, even after you adjust for the age of the other 9 organs. The state of your brain seems to be the ultimate limiting factor for your health span.
And it's linked to neurodegenerative diseases, or NDs, is maybe the most shocking part of this data?
It is. The brain age gap showed the strongest link to future risk of NDs, including all-cause dementia, where the risk increased by a remarkable 88%.
Wow.
But let's look at a specific detail from the data that really drives this home. Individuals who later developed Alzheimer's disease, they exhibited the largest brain age gap at baseline, before they had symptoms. Their brains were aging on average 2.2 years faster than their chronological age.
2.2 years.
Meanwhile, their overall organismal aging was pretty modest, only about half a year accelerated.
So long before cognitive symptoms or any typical diagnostic markers show up, this clock is telling us which system is about to fail. What molecular information is the brain clock actually using to make this call?
It gives us molecular insights by tracking key proteins associated with underlying brain pathology. For example, it detects something called glial fibrillary acidic protein, or GFAP.
And what's that?
That's a key marker of glial activation, essentially the immune cells of your brain turning on and getting inflamed. It also tracks neurofilament light chain polypeptide, NEFL, which indicates that your neurons are being damaged.
So we're literally measuring microscopic damage in real time with just a blood test. That's incredible. And this also leads to the concept of resilience, right? Which is so important for people with genetic risk factors.
Precisely. The research was able to stratify Alzheimer's risk based on those notorious APOE haplotypes. But here's the good news. They found that a super youthful brain agiotype, a brain that appears biologically much younger than expected, seems to confer genuine resilience.
How much resilience?
This youthful brain reduced dementia risk by a substantial 60%, even among people carrying the high-risk APOE33 variant. It means your biology can, in some cases, trump your genetic destiny.
And this brings us back to the most actionable element, modifiability. If lifestyle choices can accelerate the clock, then fixing those choices should slow it down.
It does. Accelerated aging, especially in the brain and the pancreas, 2 organs critical for both cognition and metabolism, was strongly associated with classic unhealthy lifestyle factors: smoking, poor diet, and, critically, not getting enough sleep.
And on the flip side,
Conversely, a robustly healthy lifestyle was linked to deceleration in those very same organs. If you want to invest in your longevity, the Pakis tell you exactly where to put your chips first.
given the potential for personalized medicine here, what are the hurdles to clinical translation? I mean, do we really need a lab to measure thousands of proteins every year?
The researchers actually preempted that concern beautifully with what they called parsimonious models. They developed a refined brain clock using only 10 non-fasting plasma proteins.
Just 10.
Just 10. And critically, this slimmed-down model retained 88.4% of the predictive performance of the full complex model. That makes the concept of integrating a brain aging measurement into your annual blood panel completely feasible.
That makes the technology feel so much closer to reality. Now let's transition from the macro level tracking this decline to the micro level, the actual cellular mechanism that causes this age-related inflammation. We're talking about cellular senescence.
Right. Senescence is where a cell, usually after some serious damage like DNA damage, just stops dividing. It's a good failsafe against cancer, but the problem is these senescent cells don't just sit there quietly.
Whoa, they get loud.
They get very loud. They start broadcasting inflammatory signals through what we call the senescence-associated secretory phenotype, or SASP.
Yeah.
And the SAP is this chronic, damaging inflammation that accelerates aging in all the tissues around it.
And the source material highlights that this SASP state is triggered by physical components inside the cell.
Yes. The inflammatory response is often triggered by pieces of damaged DNA that are literally expelled from the cell's nucleus into the cytoplasm. These are called cytoplasmic chromatin fragments, or CCF.
Okay, CCF.
And when the cell's internal sensor, a pathway called CGAA STING, detects these fragments floating around outside the nucleus, it basically panics and turns on that inflammatory ASASP response.
So now we get to the core paradox. P53 is that famous molecular police chief, the guardian of the genome, known for inducing senescence when a cell is damaged. But these new sources show the same protein actually suppresses the formation of these toxic CCF fragments and the inflammation.
Right.
How can the inducer of senescence also be the suppressor of its worst symptom?
That's the P53 paradox. And it's because P53's highest priority is genomic integrity. When it detects DNA double-strand breaks, it enters the nucleus. And instead of just triggering cell cycle arrest, P53 actively suppresses CCF formation by enhancing the cell's ability to repair the DNA damage inside the nucleus.
So it prevents the damaged DNA from being thrown out in the first place.
Exactly. Let's use an analogy. The nucleus is a fortress. DNA damage is a break-in. P53 rushes in, not just to lock down the fortress, that's senescence, but to clean up the mess and prevent the evidence, the damaged DNA, from being tossed outside the walls where it would alert the neighbors and trigger a riot, the SASP.
That's a perfect metaphor.
And this cleanup duty is heavily dependent on P53's key transcriptional target, a protein called P21. The studies confirm this. Activating P53 helped preserve the intact genome in senescent cells after stress. And fascinatingly, they looked closely at the garbage that was tossed out at the CCF.
And what did they find about the fragments themselves?
They weren't just random bits of broken DNA. They rarely included centromeric sequences, but about 80% of them contained telomeric DNA.
The ends of the chromosomes.
The ends of the chromosomes. It suggests that damaged DNA from the telomeres is preferentially expelled when the cell decides to ditch the evidence. It's like forensic science at the cellular level.
So if P53 activity is the key to creating a quiet, non-inflammatory, good senescent cell, how do we boost that activity therapeutically?
This leads to a strategic shift in anti-aging research. Instead of trying to kill the senescent cells, which is what senolytics do, we're now looking at xenomorphics, interventions that change the behavior of the senescent cell.
Okay, xenomorphics. And the source material focused on using pharmacological MDM2 inhibitors, right? MDM2I.
Correct.
For our listeners who might not be deep in molecular biology, what is MDM2 and what does inhibiting it actually do?
MDM2 is essentially the protein that marks P53 for degradation. It acts like the regulatory brake on P53.
Yep.
So when a cell senses damage, P53 activity goes up, but MDM2 is always trying to bring it back down.
So you inhibit MDM2.
You inhibit MDM2 with a drug like HDM201, which they used in the study, and you release the brakes.
Yeah.
You are dramatically increasing P53's stability and its ability to do its cleanup job in the nucleus.
So the MDM2I treatment successfully ramped up P53. It suppressed the CCF formation and it dampened those inflammatory SSP pathways. And this is the critical distinction. This was a xenomorphic intervention.
Exactly. It was confirmed to be not senolytic. The drug didn't kill the senescent cells. It didn't reverse the cell cycle arrest. Its entire purpose was just to turn off the inflammatory megaphone those cells were holding. This is a huge strategic shift.
Why is it so huge?
Because it avoids the potential complications of killing off senescent cells that might actually be needed in certain tissues. You're just mitigating the damage they cause.
So did this promising strategy actually work in a living organism?
Yes, they tested it. They used the MDM2I, HDM201, in naturally aged mice over just 14 days. And the results were quite compelling, particularly in aged female mice. The drug reversed a clear age-associated transcriptional signature. It significantly downregulated key immune and inflammation genes, like TNF and interferon gamma, and it upregulated beneficial metabolic genes. It also reversed the age-associated accumulation of inflammatory immune cells, like macrophages, in the liver. It's a clear reduction of systemic inflammation.
That's a powerful result. But let's tie this back to the cell's powerhouses, the mitochondria. We know mitochondrial dysfunction drives aging, but how does it influence this delicate P53 and CCF circuit?
This is maybe the most nuanced finding, and it provides the ultimate connection point. We know mitochondrial dysfunction causes stress, which leads to CCF formation and SASP, but here's the surprising twist they uncovered.
Okay.
When researchers experimentally ablated or destroyed the mitochondria in senescent cells, two things happen. First, it stopped CCF formation. Second, it paradoxically activated P53.
Wait, that's counterintuitive. Why would removing a driver of damage, the dysfunctional mitochondria, also activate the key protective protein, P53?
Because the damaged DNA, the CCF, could no longer be expelled from the nucleus. The dysfunctional mitochondria were essentially acting as an expulsion mechanism. By driving the expulsion of damaged DNA, the dysfunctional mitochondria were simultaneously lowering the DNA damage burden inside the nucleus.
I see. So the nucleus looks cleaner than it really is.
Precisely. If p53 is the detective sensing DNA damage, the dysfunctional mitochondria were basically pushing the crime evidence out of the house. This gave p53 a false sense of security, preventing its full activation.
Then allowing the cell to become chronically inflammatory.
Exactly. So when you remove the mitochondria, the damaged DNA is retained in the nucleus, the damage signal spikes, p53 gets fully activated, and the cell is forced to initiate its non-inflammatory protective measures.
That is a phenomenal piece of insight. It just ties the macro-level tracking of organ decline right down to the micro-level failure of cellular defense. So we've established that these proteomic clocks can validate organ-specific aging and predict disease years out, particularly in the brain.
And we've found a critical, manipulatable cellular circuit. p53 is the defender of the genome against inflammation, but its effectiveness is being undermined by dysfunctional mitochondria that are driving the expulsion of damaged DNA. This means we can either use senolytics to kill the cell, or, much more precisely, xenomorphics like MDM2 inhibitors to force p53 to clean up the genome and shut down the SESP.
So for you listening, looking to maximize longevity and health span, what is the core takeaway from this deep dive?
Well, that the brain age gap is the single strongest predictor of mortality. And we know that unhealthy lifestyle factors accelerate that gap. Then the future of anti-aging isn't necessarily about blanket interventions, it's about precision.
Right.
We now have the tools, these PACs, to identify exactly which of your organs is failing fastest. And we also have the specific molecular strategy, the xenomorphic approach targeting p53, to quiet the destructive inflammation that accelerates that decline.
Which raises a final provocative thought. If the brain and pancreas are the most modifiable and accelerated aging in those two organs leads to the most catastrophic outcomes, does the true path to longevity lie in highly targeted, organ-specific lifestyle changes and eventual xenomorphic drugs designed explicitly to protect these systems? Turning off the cellular megaphone before it even starts shouting, that's knowledge you can definitely use.